Guys, let's continue the gene transfer methods in today's lecture. Second gene transfer method is transduction. We have seen transformation in detail. So complete video on this transformation. The link for this video will be uploaded in my description box. Please do check out. So transduction is gene transfer method in which DNA fragment of one bacterium transferred to other bacterium by bacteriophage. That means bacteriophage helps in the gene transfer method. Okay. So transduction generally carried out by two different methods, that is generalized transduction and specialized transduction. Okay, so basically bacteriophages, we have seen bacteriophages are the viruses which kills bacteria or eats bacteria or lice bacteria. Okay, so generalized transduction is carried out by lytic or virulent bacteriophage, while specialized transduction is carried out by lysogenic or temperate phage. Okay, because bacteriophage have two life cycles or two replication cycles, that means lytic cycle and lysogeny. This lytic bacteriophage target any donor gene, while lysogenic bacteriophage targets specific donor genes. In this generalized transduction, bacterial cells rapidly lyse, that means degraded or denaturated, you can say. And in specialized transduction, bacterial cells cannot lyse easily and can survive for a longer time period. In generalized transduction, bacteriophage enclose a part of donor DNA inside capsid, so it is called as prophage. And in specialized transduction, during the detachment process from bacterial chromosome, small amount of bacterial DNA remain bound to viral, that means phage DNA, and thus continues in new phage capsid. This is the major step in specialized transduction. We will see in detail. In generalized transduction, viral DNA does not incorporate into the bacterial DNA, we have seen already. And in specialized transduction, viral DNA and bacterial DNA combines, or you can say integrates. So let's see the first one, generalized transduction. As we know that generalized transduction is carried out by lytic bacteriophages, and gene transfer method mainly involved recombination process. We know that recombination means joining and attachment of donor DNA and recipient DNA, right? So in first step we will see step by step how generalized transduction is carried out. In first step, phage infects the donor bacterial cell. In this, phage first of all attach to the bacterial cell. That means first one is the attachment process, you can say. Bacteriophage gets attached to the bacterial cell, then phage DNA and proteins are made and bacterial chromosomes is broken down into pieces. It means that in first step phage DNA enters inside the bacterial cell, then using the host cell machinery, bacteriophage what will do, it will degrade the bacterial chromosome. Okay, bacterial DNA, you can say sometimes plasmid or bacterial chromosome gets degraded using bacteriophage DNA via host cell machinery. Okay, in the third step, occasionally during phage assembly, that means now using host cell machinery, phage will produce its capsid coat proteins and DNA. Okay, because it is intracellular parasite, we know that bacteriophage is intracellular parasite. Using host cell machinery, it will, what will happen, it will produce its assembly, and occasionally sometimes mistakenly what will phage do, phage will do as piece of bacterial DNA are packaged in phage capsid. So sometimes mistakenly क्या हो जाता है, phage rather than using its DNA, its replicated DNA, it coats on encodes bacterial DNA inside its capsid. Okay, then donor cell lyse and release phage particles containing bacterial DNA. Then after the cell lysis, bacterial DNA containing bacteriophage will lysed. Then again this phage having bacterial DNA will infects new host cell, that means recipient cell, and thus recombination takes place, in which donor bacterial DNA and recipient bacterial DNA gets combined or recombined, you can say. Thus recombination can occur, producing a recombinant cell with a genotype different from both the donor and recipient cell. Okay, thus at last, during using recombination, gene from the host donor cell, that means host bacterium cell, transferred to the recipient cell. So in this way, generalized transduction is carried out. Examples are Salmonella, Escherichia coli, Staphylococcus and Pseudomonas. In this bacterium, generalized transduction mainly takes place, and one example is that Staphylococcus has a penicillinous coding plasmid, which is transferred from one Staphylococcus to another Staphylococcus using bacteriophage by the process of generalized transduction. Now coming to the second one, specialized transduction. We know specialized transduction takes place or carried out by lysogenic bacteriophages, or you can say non-virulent bacteriophages. This process occurs occasionally, that means हर बार specialized transduction नहीं होता है, most of the time generalized transduction ही होता है. So lysogeny, that means the in favorable conditions, lysogeny takes place by temperate bacteriophages. Okay, so in this similar process takes place. First one is that bacteriophage first of all get attached to the host bacterial cell, integrates its DNA inside the bacterial cell. Okay, then phage DNA integrates into the chromosome. Phage DNA integrates into the chromosome, and this is the major step of specialized transduction, where prophage is generated. That means phage DNA is attached to the bacterial DNA. This stage is nothing but the prophage stage. Okay, then in the next step, prophage deintegrates and mistakenly picks up piece of bacterial chromosome. You can see in this diagram, this prophage mistakenly take up the bacterial chromosomal DNA, and thus replication of viral DNA and destruction of bacterial DNA takes place. That means using host cell machinery, this prophage containing bacterial chromosome will generate or replicate viral DNA and responsible for the production of virus capsid and synthesis and assembly of the phage proteins and phage structure. Okay, then cell lysis takes place when favorable condition occurs, or rather than we have seen that cell can survive for a longer time period having prophage inside it. Okay, then after the cell lysis, crossover and stable gene transfer into recipient cell chromosomes takes place. That means again this prophage will transfer to the recipient cell having the bacterial chromosomal DNA along with the phage DNA. Okay, so in this way specialized transduction takes place. So this is the gene transfer method of transduction. Thanks for watching.